So again, through the serial deletion of these tubes, your assay sensitivity will be determined. So negative control where there's no endotoxin present, and then diluted amount of endotoxin slowly going down its concentration. Okay, to all these tubes, you will add the same amount of LAL solution. Okay, to all these tubes, and you'll expect to see some gel formation because some of these tubes should have enough endotoxin that will form gel, but this one should not because there's no endotoxin present. Okay, so let's say based on that, okay, you detected gel formation from these tubes. Let's say, the negative control, there was no endotoxin, there's no gel formation because there's no coagulation happening. But, from these tubes, okay, like a 0.5 endotoxin unit per mil, 0.25 endotoxin unit per mil, 0.125 endotoxin unit per mil, you are going down with the endotoxin concentration because they are serially diluted, right? You saw that, gel formation from maybe first 3 tubes, okay, but there was no gel formation from these last 2 tubes because the amount of endotoxin present there is very small, so it's not good enough to form any gel. Then from this data, what you can tell is what? You need at least about 0.125 endotoxin unit per mil of concentration to be able to form gel in your assay, right? So that will be the assay sensitivity that is determined by your first set of experiment. Of course, this is an example. Your result may be a little different than this. Okay, I'm just giving you an example. So if you get a data like this, okay, you get gel formation from the first 3 tubes with high endotoxin concentration, but no gel formation from these 2 tubes with the weaker endotoxin and no endotoxin present there. That means the least amount of endotoxin that will be detected as a gel formation will be 0.125 EU per mil. That will be the assay sensitivity, okay, that you can determine from the first set of the experiment. Does it make sense? Okay, once you have done that, now you can do your test. Okay, you'll be given with a water sample. It could be a tap water sample from the faucet in the lab, or it could be a deionized water sample that you make in our building, or it could be the purest water we have. It's a nano-pure water. Okay, whatever the water sample you're going to test, you will set up a series of the tube, kind of similar way. Okay, but here instead of using those endotoxin, you're going to use your testing water, okay, to make a serial dilution. Okay, so like the previous case, the first tube will have no testing water. It's going to just have endotoxin-free water, and then you're going to add LAL to it. Okay, but these tubes will have your testing water, maybe a diluted testing water, okay, 1-to-1 dilution. Okay, and then from that test tube, you're going to dilute it further by transferring half of that to the second tube, and that will be mixed up with endotoxin-free water. Okay, and then you continue that dilution one more time. Transfer half of it and mix with the endotoxin-free water, and transfer half of it, mix with the endotoxin-free water. You continue that one more time. So then you will see the amount of testing water, maybe it's a tap water or deionized water or nano-pure water, whatever you are testing, that will be diluted continuously, or half concentration, a quarter concentration, or eighth concentration, or 16th concentration, or 32nd concentration. Is going to go down continuously, right? And to those tubes, you're going to add LAL, the same amount, okay? And then you're going to see if you will detect some gel formation from these tubes, right? So let's say when you did that, you got this result. Okay, so negative control, there's no testing water present. So since it's given with the no endotoxin free water, so of course it's not going to form any gel when LAL is added. But from these tubes where there's enough testing water is present, maybe a half dilution, fourth dilution, eighth dilution, 16th dilution, you see the gel formation. But the last tube, you got maybe 1/32nd dilution, there was no gel formation. Now what does that mean? So that means these tubes had enough endotoxin, so they gave you a gel formation. Okay, if you look at the trend, okay, so that will be the least amount of endotoxin, okay, that gave you a gel formation, which is similar to what you got here. Okay, so that was the least amount of endotoxin that gave you gel formation, which was what? 0.125 EU per mil. Okay, by looking at that trend, you can say the same thing. If you look at the trend, maybe that tube has the same amount of endotoxin like this case, okay, which is 0.125 EU per mil. You can assume that yes, this tube has that 0.125 EU per mil endotoxin present there, right? So that's what you can expect from that tube. Not only that, so in that tube, how much of the testing water is present? Okay, it's present at 1/16th dilution, right? If you know that, then how about the undiluted amount of water? Let's say you test the tap water. Okay, how much of endotoxin will be present there? Okay, if the tap water's 1/16th dilution gave about 0.125 EU per mil, okay, then the undiluted straight tap water should have about 16 times more endotoxin present there, right? So you just multiply the dilution factor to that assay sensitivity, then that'll give you the amount of endotoxin present in the undiluted testing water. So that's how you're going to calculate out the endotoxin present in the testing water. Does it make sense? Okay, if it doesn't make sense, you can play this over and over. Okay, so that's how you're going to test the endotoxin amount present in your testing water. That's going to be the second lab exercise, and then based on this result, you will write your second lab report, which is about 25 points. Okay, you will write it up and upload it on your course shell. Okay, and then third part of the lab, it's about the autoclave validation. So autoclave is a what? It's a machine that can sterilize your materials, solutions, or any kind of instrument that you need to sterilize. You can use this machine. Basically, autoclave is a what? Pressure cooker that will provide your chamber with very much heated pressure steam that can kill any kind of bacterial or contaminants that may be found from your material, right? By placing your solution, or by placing whatever the instrument you want to use within that chamber, autoclave chamber, and close the door, and then introducing high pressurized heated steam into the chamber, you can kill all the bioburden, the contaminants within the chamber. So you provide this condition. About 5 to 20 pounds per square inch of the space, that's the pressure given within your autoclave chamber. And then the temperature is about 121 degrees Celsius, plus minus 0.5 degree. So that's very hot temperature. Under the condition, if you keep the condition about 15 minutes, almost all the things will be killed, right? So that's how you can sterilize things. So autoclaving is one way of sterilizing materials, and there are different ways besides this. Maybe gas sterilization is another way, and strong light exposure, that's a light radiation based sterilization. There may be another way, maybe you can use something like filtration if your material is heat sensitive. Okay, so anyway, so you're going to use an autoclave today to sterilize the solutions that you will be making. 10x TE or 10x TA you're going to make. You're going to put that in a bottle, and you're going to sterilize the solution using an autoclave. Okay, so then how do you sterilize it? So you're going to place your material, the solution, in a bottle inside of autoclave, and then you're going to shut the door and then turn on the autoclave. We're going to talk about that. And then when you use your sterilizer, the autoclave, you need to validate the function of the autoclave. That's validation is always the CGMP requirement. Okay, any kind of machine, instrument you're using, that machine should be validated. We talked about what? IQ, OQ, PQ qualification method that qualifies the instrument, as well as the validation is what? The qualifying the process is a validation, right? So how do you know if your autoclaving cycle, the process really indeed kill all the contaminants or not? So that's why we consider it as a validation, autoclave validation. How do you do that? Okay, there are many ways of validating your autoclave cycle, but among those different ways, there's a way of using a simple bioindicator ampules. Okay, we have some bioindicator ampules in the lab. Okay, those ampules have special bacterial cells. Okay, they have the extreme thermophiles that can withstand high temperature conditions. Okay, so those ampules will have the spores of those thermophiles. Okay, if you place those bioindicator ampules that have these thermophiles, Geobacillus, Thermophilus is the name. Okay, they can withstand the high heat. Okay, but they cannot withstand the autoclave cycle, the validated cycle. But if the cycle is not maintained for the right amount of time, if the temperature is not correct, these spores can survive through your autoclaving cycle. Okay, so by placing the ampoule in your autoclave cycle, and then at the end of the autoclaving cycle, you're going to take the ampoule out and then see if you can have those bacterial cell spores come back to life. Okay, by placing that in an incubator that's set up at the optimal temperature condition. Okay, if you can see the survival of these spores back to the bacterial cells, that means what? Then your autoclaving cycle did not kill the cells, so it is not validated. So that's how you validate the autoclave cycle, simply by using this autoclaving ampoule. So that's what you're going to do. Okay, if the autoclave did its job, of course you will kill the spores, and spores will not come back to the life. Then the ampoule will not be turbid because of no bacterial cell growth. If bacterial cells are not growing, then there will be no color change of solution. There's a pH indicator in that ampoule. It comes as in purple, magenta color at the beginning, but if it's growing the cells, and cells will eat up the medium, and then they will change the pH. When the pH drops, that purple magenta color will become yellow color. So if you see the color change, that means there are cells growing in there. That means your autoclaving didn't do its job, so it didn't kill the spores. So your autoclave is not validated. So that's how this bioindicator works. Okay, so hopefully you will get to use that and see the result at the end of this lab. All right.